lesson 9 group problems. So problem 1 was to diagram the intermediate stage of DNA replication for bacterium E. coli. And so you can see I've drawn it here on this paper. And this is just the codes for the different things on there. This would be the ORIC, the origin of replication right there. And the bacterium E. coli just has the 1, and it kind of splits off. The strand kind of separates there. And you have your leading strands and then your lagging strands, which have the primase, a different primase, and then the primase for a leading strand. So that way they can start off the replication. The DNA helicase you will find at the fork because it is unwinding the DNA. And then the Okazaki fragments are combined of the primase and the lagging strand. I think the DNA polymerase III alpha subunit just comes off and helps bind the different ones. And so, yeah, those are the different ones that are indicated in the drawing.
Sweet. Okay, going to number 2. This is our wonderful vocab page, or important terms to know, and so it's filling in the blank. So for letter A, the protein complex that synthesizes primers in eukaryotes. Obviously, that is the DNA primase. Important role there. For B, the protein promotes the separation of the AT-rich sequence with the origin of replication. That's helicase is kind of like acting like a wedge to help that separation, separating the double helix. So the subunit of DNA polymerase III that contains the proofreading activity, that's epsilon. That's the epsilon subunit of DNA polymerase III. Kind of a cool little molecule. Does a whole ton of stuff because there's almost an entire Greek alphabet of DNA polymerases out there. So letter D is talking about this protein synthesizes the leading strand in eukaryotes. That is DNA polymerase III, the workhorse of DNA replication. Very important molecule to understand and know. For letter E, the protein that displaces DNA polymerase III from the chromosome to terminate replication are TUS proteins. For letter F, the protein that has 5' to 3' polymerase and 5' to 3' exonuclease activity. So that's DNA polymerase I. Not as a big hitter, but still an important role. So letter G is talking about the protein that introduces negative supercoils into DNA ahead of the replication fork, making it easier to separate. That's DNA gyrase. So letter H, this protein binds directly to the ARS elements. That's the pre-RC protein. For letter I, the protein that prevents DNA strands from annealing during replication. That's the making sure that that the proteins and enzymes get to the right place. Those are the single strand binding proteins. And the last one, letter J, is talking about the enzyme that seals Okazaki fragments into a continuous strand of DNA. So in replication, because from the 5' to 3', I may have this backwards, but anyways, the 5' to 3' strand it's almost continuous. Like you don't have to. It's being replicated continuously by DNA polymerase III off of an RNA primer. And now the reason why we have DNA ligase is because we got all these DNA Okazaki's fragments piggybacking from one primer to the other, because DNA, this wonderful molecule has figured out some way to go in the reverse direction. So you need DNA ligase to help bind all of those together.
Alrighty, so problem 3 is just like another term. So enzymes that consume ATP during DNA replication would be DNA primase, DNA ligase, DNA gyrase, and DNA helicase. Enzymes that consume DATP would be polymerase 3 and polymerase 1. Enzymes that have a 3' to 5' exonuclease activity would be DNA polymerase 3, DNA polymerase 1, and telomerase. An enzyme that would be involved in replication that uses DNA as a template to synthesize RNA would be primase. We know that that is its function. One enzyme that uses RNA as a template to synthesize DNA would be DNA polymerase 3.
Sweet. Sweet, sweet, sweet. So number 4 is this wonderful application of what we're learning about, which is really kind of neat because in my research group we were tasked with the opportunity to run a PCR reaction. PCR reactions, I guess, is actually another kind of offshoot of what we're doing here in number 4. It's number 4 is basic. PCR is it's a chain reaction where we generate primers to generate certain strands of DNA and amplify that a billion times. You stick your primers and your DNA sequence in a little microfuge tube, and you stick it in this little machine called the thermocycler, and it runs these cycles simultaneously for a long time, and you can generate a lot of copies of one type of gene. And so I got to learn about this in a little bit more detail, and I thought that was really cool. So number 4 is like real life application. Like this is important stuff, and I thought that was the coolest part. So back here in showing my work right here. Here is number 4. So they give us the sequence we want to replicate, and then they give us the primer, which is super nice. Because if your research mentor is like, hey, we want you to take this gene right here, like for example, GAPDH in zebrafish is what I'm doing. Like, here's your GAPDH gene. I want you to find a primer. So in many cases, they won't give you the primer, and they'll say, okay, here, just go find in the scientific literature, find a primer that'll work for this gene. And I'm like, okay. But that's nice. Number 4, they gave us the primer right here. So the deal is we have to find out the complementary part of this gene that primer will bind to. And so we have our directionality of this gene right here is 5' to 3'. And now we look on our primer here, and because it has the complement, it flips, so it goes 3' to 5', whatever your direction you're looking for, wherever this primer will bind to. Next thing we're looking for is at this 3' end, we're looking for the sequence that will stick to the other one. So in this case, G goes with C, G goes with C, A goes with T. So we probably know that this primer is going to attach this way. So this primer attaches this way. So G to C, G to C, A to T, A to T, A to T, A to T, T to A, T to A, G to C to G, G to C, T to A. Bam, there it goes. Okay, for letter A, it's asking, whoa. For letter A, it's asking, so the nucleotides in this reaction include DATP, DCTP, DGTP, and DTTP. So it's saying that this deoxyadenine triphosphate, deoxycytosine triphosphate, deoxyguanosine triphosphate, and deoxythymine triphosphate. This means that we're adding these nucleotides and that they have the right amount of ATP associated with them that they can add to the strand. So back in that work problem, like showing my work here, because these nucleotides have the sufficient amount of energy to bind, they will all bind. So from the 5' to 3' directionality, this is what the sequence will be. And they asked us not to include the primer sequence, so boop, we cut it off right there. That's the sequence. For letter B, it's asking us the same deal here, but it's saying that we have deoxycytosine monophosphate. Not enough energy for the cytosine molecule to add. So from over here, we're adding this way. We're going 3' to 5', boop, adding this way. We can add an A, but we can't add a C because we don't have that nucleotide doesn't have enough energy to bind to the strand. So replication stops at where you get the C. Boop, stops right there. So you get 5' A, 3'. That's it. Letter C is talking about the same deal here. We have all of the other necessary nucleotides, but excluding T. So we get UTP. That means we're doing RNA then. And so we go up to where we get a T. So we get an A, we get a C, and then right here at A, if we had that deoxythiamine triphosphate, we would add it, boop, right here at A. But we don't. We have the uracil triphosphate, and we're not doing RNA in this moment. So our sequence stops at AC, 5' to 3'. So D is the same deal. It's the same deal. I think I misplaced the wrong. And D is talking about, right, okay. Right. So the same deal there on my sequence, you'll stop at where you need G. And so you get ACT, 5' to 3'. And that's how you do that gene replication problem.
Okay. So problem number 5. The proteins that are found in prokaryotes are TUS, DNA-A, DNA polymerase I, DNA polymerase epsilon, DNA primase, MCM helicase, and DNA gyrase. I was not able to find B. So we'll move on to C. The proteins involved in compaction would be DNA polymerase alpha subunit, DNA polymerase III alpha subunit, and pCNA. The initiation stage of DNA replication involves CDC6, and the protein for termination of DNA replication would be TUS. Okay, and that concludes our problem.